Welcome back, dear students. Let's take our biotechnology lesson forward with our third lecture of biotechnology. Just a quick recall of what we have tried to understand in biotechnology till now. In lecture 1, we basically discussed the introduction. What exactly is biotechnology, its type, traditional and modern, and in modern biotechnology, what we try to do genetic engineering, that is modify the genome of an organism to make it beneficial for humans, and of course trying to maintain sterile or contamination free conditions to maintain the culture of that genetically modified organism. Then in lecture 2 I basically told you about the most basic point of modern biotechnology, that is first of all identifying your target, what you want to do. For example, I want to actually help the humanity overcome the menace of diabetes mellitus. Diabetes mellitus is the disease that is caused by insulin deficiency. So if I want to actually treat diabetes mellitus, what would I require is basically a desired product, who is that insulin, or maybe any other desired product, right? If I want a desired product, obviously I have to identify a desired gene that is going to code for this insulin or any product of my choice. Then I also have to identify an organism who is actually carrying this gene, because I could have obtained the product from the organism itself only, but the problem is the organism is making the product for its own use. It is not making the product for human use. It is not making the product at a commercial scale. I would require insulin or any other protein product for that matter that I want to help the humanity with to cure diseases. I would want that product at a mass or a commercial scale. So to get a product at a mass or a commercial scale, I would have to produce that product. But how would I produce that protein product? With the help of a gene. So how would I get the gene? Obviously I would have to identify an organism who carries this gene, okay, and then isolate or obtain the genome of the organism. Because it's impossible to carry out to take out a single gene from the organism, so we would basically isolate the genome of this organism. The genome isolation process I told you in the previous class, that ended with the step where in the test tube where we had taken the cells of the organism whose genome is to be isolated in a buffer solution. First of all, we added the cell wall digesting enzyme if you are taking out the genome of a plant, fungus, or bacteria cell. And then you basically add cell membrane digestive detergents like SDS if you are using an animal cell, or let's say if you are trying to take out the genome of an animal cell, you just simply do the treatment with SDS, sodium dodecyl sulfate detergent, that digests the cell membrane, and then you basically do addition of chilled ethanol. The addition of absolute chilled ethanol had resulted in creation of precipitates of DNA in this solution. Okay, so these were your DNA precipitates representing the genome of an organism who is the natural source of a protein product which we want to produce at a mass or a commercial scale for human benefit. So this, if you have understood, is the entire story in biotechnology that we have discussed till now. I want a protein product at mass scale for human benefit. But then option 1, I can get that protein product from some organism who is producing that protein product. But in how much quantity that organism is producing that protein product? Limited quantity for its own use only, not for human use. So if I have to actually produce that protein product at a mass scale, I would need that gene which is coding for that protein product. And to get that gene, I'll have to isolate the genome of the organism. So this was our entire story in the first two lectures: introduction, what we are trying to do, and how to isolate the genome of the organism. DNA precipitates are formed. The most important question, as I told you, from DNA precipitation process: DNA precipitates formed after addition of which reagent? Chilled absolute ethanol. And this DNA is exactly basically what? The genome of the organism, who is the natural source of the protein product that we want to produce at a mass or a commercial scale. So now what we need to do today is basically, first of all, from this test tube, separate these DNA precipitates. So what will these DNA precipitates be? They will be the genome of the organism, who is the natural source of your desired protein product. So inside this genome, that gene also will be there, which is going to code for your desired product. So let's first take out this genome from the test tube and then try to break down this genome into smaller pieces. What will those smaller pieces be? Genes. And then try to identify that particular gene which you want, or the gene that is going to code for your protein product. So this is all our lecture number 3 is about: how to take out these DNA precipitates or the genome from the test tube, and how to break it down into smaller fragments or the genes with the help of enzymes, as the lecture heading says, restriction endonuclease enzymes. So well, let's begin. The DNA precipitates which are obtained in the test tube by adding chilled absolute ethanol, the DNA or the genome precipitates formed in the test tube by adding what? Chilled absolute ethanol. So this is, as I told you, the most important question from the genome extraction process: addition of which reagent is responsible for formation of precipitates of the DNA? Chilled absolute ethanol. Okay, so the DNA or the genome precipitates formed in the test tube by adding chilled absolute ethanol first are extracted or separated from the rest of the solution. What exactly is the rest of the solution? As you might recall, when we started with the genome extraction process, we had dissolved a few cells of the organism whose genome is to be isolated in the buffer solution. Then what did we add? Cell wall digesting enzyme or cell membrane digesting detergents like SDS. Then we basically add RNAs and proteases also to digest the RNA and the proteins which are present inside the cell here. And then finally we added the chilled absolute ethanol that resulted in precipitation of the DNA strands. Okay, so now the rest of the solution is basically what? The broken down components of the cell wall, cell membrane, proteins, RNA, and the only thing that is intact right now is basically what? DNA in the precipitate form. So I need these precipitates from the rest of the solution. So how do I separate them? One method that I told you that day was, take a glass rod and put it in, and with the help of this glass rod, which may be basically having a hook like structure at the end, you can simply lift or pick out all the DNA fragments. So taking out the DNA precipitates with the help of a glass rod, this is a manual technique that is called spooling technique. The DNA or the genome precipitates formed in the test tube by adding chilled absolute ethanol are extracted or separated from the rest of the solution by either spooling or by, of course, the other possible method is centrifugation. Centrifugation is done at a gentle speed in this case, And when you do the centrifugation at a gentle speed, obviously you can see the logic that all the DNA precipitates are going to settle at the bottom, and we can just throw the rest of the stuff. Okay, so DNA precipitates can be extracted by centrifugation at a gentle speed so that the DNA precipitates settle down at the bottom of the test tube, and the rest of the stuff can simply be discarded. The remaining solution that stays on top, that can be discarded. So this is one method, and this is obviously a more efficient method, because centrifugation at a gentle speed would actually help your DNA precipitates to settle at the bottom. You can just throw or discard the rest of the solution. Done and dusted. You finally have the DNA fragments in your test tube. The other option is of course more tedious because it's manual. It will require precision. Okay, spooling is putting in a glass rod in this test tube and rotating the test tube, winding the DNA fragments on the glass rod. It's like basically simply lifting them. It's like, something has fallen in your tea coffee mug, and you take a spoon to, take out that thing, the foreign thing. Okay, so you just take a spoon and scoop out whatever you want to. So similarly, just take a glass rod and with the help of it, scoop out the genome or the DNA precipitates. So whatever method you use, if you are basically, let's say, a poor scientist who cannot afford a centrifugation machine, we can always do spooling. Centrifugation machine is available. Of course, this is an easier and a better choice. Okay. So well, anyways, in any case, at the end of both these processes, I would basically have a test tube in which I would be left with what? Only the DNA precipitates. And this is not just any small DNA fragment or whatever, it's actually the entire genome content of the organism who is a natural source of our desired protein product. Okay, so let's move on to our today's step here now. We've got DNA precipitates, the entire genome content of the organism who produces our desired product, but for its own purposes, not for us. So once I have got the DNA precipitates over here, the entire DNA precipitates or the genome is obviously of no use to me. What do I need is a small fragment, a small gene. So what should I do now? I should basically try to cut or digest these DNA precipitates, break them down into smaller fragments, and then identify that particular fragment or the gene that will code for my desired product in future. Okay, so once I've got the DNA precipitates available here, the entire genome content, I should add some buffer to keep it hydrated, obviously, because I cannot keep the DNA dry for a lot of time because it is going to get denatured. The DNA precipitates or the entire genome content is kept in another test tube and let's say if I've taken out the DNA precipitates by spooling, I would basically collect them in a separate test tube, and small amount of buffer is added to the test tube to keep the DNA in hydrated form. Okay. So right now I have got the entire genome of an organism. Which organism? The one who's a natural source of my desired protein product. Okay. So my next aim is now digesting or breaking down this genome. This genome needs to be digested or broken into what? Fragments. Fragments means the genes, so that I can get that gene which will code for my product, and then use that gene further in more steps. Somehow we'll also discuss that also, how to use that gene, and finally obtain the desired protein product at a commercial scale, making everyone happy. Yourself happy because you can sell and earn, others happy because they can buy and get rid of the disease. Okay, so this genome needs to be digested or broken into fragments, and to digest a genome, what do you require? Enzymes. The digestion process obviously needs enzymes. What kind of enzymes digest the genome or the DNA? What are they called? The enzymes which digest DNA or the genome which is there in the test tube, what are they called? Nucleases. The enzymes which digest the genes or DNA or a genome, what are they called? Nucleases. Okay, now if you talk about nucleases, where do we get them from? See, we've got a test tube with the genome in it, and I need to digest this genome, so I need to add the enzymes in this test tube only. Okay, so if I have to add enzymes in this test tube, I need enzymes. Where do I get the enzymes from? Somewhere. That is what we're trying to understand here. Okay, the enzymes that you will need to digest the DNA or the genome, they're called nucleases, and some of the nucleases are found in the human body also. We also have nucleases produced by our pancreas, organ of the digestive system. We also have nucleases produced by our pancreas, but can we take out these nucleases from our pancreas and put them in the test tube over here to digest the genome? Actually, unfortunately, no. We also have got nucleases produced by our pancreas, but the problem with these enzymes is that these are exonucleases. Exonucleases means if I'm talking about the genome that I need to digest, just a diagrammatic representation, let's say this is maybe the genome that you want to digest. This is the representation of these precipitates, the genome that needs to be digested. So here's, let's say, a representation of that genome. 5' end, what are all these things? Nucleotides. How are they attached to each other? With the help of 5', 3' phosphodiester bonding. The other strand also same. There will be 5', 3' phosphodiester bonding between the nucleotides. So let's say this is the genome to be digested. Genome to be digested. The one that is actually available inside the test tube, the one that I've kept inside the test tube, it is that genome that I need to digest. And I've just mentioned that we also have nucleases produced by our pancreas. So let's say I actually add the pancreatic nucleases in the test tube. The problem with the pancreatic nucleases is that these are exonucleases, which means they attach to the 5' ends of both the strands. Exonucleases क्या करते हैं? They are enzymes that will attach to 5' end of both the strands. This is one enzyme that is attached here. This is the other enzyme that is attached over here. And once they attach, they will basically begin digesting the phosphodiester bonds one by one. next one, next one, next one, and so on. So we can notice what the enzyme is doing. It has started from the edges, from the outside. Now does it explain the name of the enzyme, exonuclease? Yes, because where did the enzyme begin the digestion from? From the outer edges or the outer ends. So we also have got nucleases in our own body available, but those nucleases are exonucleases. They attach to the 5' ends of both the strands and digest each and every individual phosphodiester bond. So these enzymes will be digesting each and every individual phosphodiester bond. What is the result? Are you going to get any gene fragments, or are you going to get nucleotides? We are going to get nucleotides. Here. Where is the genome now? Totally digested. And what have I got finally? Nucleotides. What have I done? Wasted everything. I actually did a lot of procedure to obtain the genome, and I just wasted it because I broke it down into what? Nucleotides. What will I do with these nucleotides? What product can I get from these nucleotides? Nothing. न्यूक्लियोटाइड से किधर प्रोडक्ट बनेगा? कुछ नहीं बनना. प्रोडक्ट तो जीन से बनना था, जीन तो बनाया ही नहीं. वो डीएनए को ब्रेक डाउन करके क्या बना दिया? न्यूक्लियोटाइड्स. सो वेस्ट कर दिया पूरा एफर्ट. सो ये नहीं करना है. Which means this entire discussion was to help you understand कि nucleases human body के अंदर भी मिलते हैं pancreas में. We have got exonucleases, but can we use them in biotechnology in laboratory? Of course not. They attach to the 5' ends of both the strands and digest each and every phosphodiester bond, producing individual nucleotides. Complete digestion that we require in the digestive system of the body. That is why pancreas is producing these enzymes over there. But the problem over here is in biotechnology, we did not need the nucleotides. We wanted genes, small sequences of nucleotides. The nucleotides cannot code for anything, which means they are of no use at all. As they cannot code for any product. So what did I learn from this entire discussion? That I would require enzymes to digest the genome. Those enzymes are called nucleases, and one of the choices is exonucleases. That is ruled out. I am not supposed to use these enzymes. The nucleotides are of no use as they cannot code for any product. Hence, exonucleases are not the preferred enzymes in biotechnology. Forget preferred, they are not used actually, because we are not going to get any gene, and without a gene what will we do in biotechnology? Nothing. So they are not preferred, or they are not the preferred enzymes in biotechnology. Not used at all. Because we don't want to break down the genome into nucleotides. We wanted to break it down into small gene fragments. Okay, so this tells us that I had an option of using some enzymes which I could have obtained from my own body, but then they are ruled out. Those enzymes are not to be used. Okay, what should I use then? I have to find some other enzymes. If exonucleases can't be used, I have to find some other enzymes. So we need to find some other types of enzymes for this genome digestion step. This step can be called the genome digestion step. Like the first step was doing your homework, deciding what you want to produce. Step 2 was your obtaining the genome. Step 3 is what we are doing now, trying to break down the genome into smaller fragments. So for this step we need some other type of enzyme, because we have learned that exonuclease doesn't work. Okay, so we need to find some other type of enzyme for this genome digestion step. Where do we find such enzymes from? We require enzymes that can attach to the genome and break it down at maybe irregular intervals. Regular intervals doesn't matter. I need enzymes that can attach maybe over here, cut, maybe over here, cut, so that I can get 1, 2 and 3 fragments. I can get 3 fragments, so that one of them maybe possibly is going to code for my desired gene. So this is something that I want, this kind of enzymes, those who can attach somewhere in between, and those who can basically digest some fixed phosphodiester bond, so that I will actually get fragments of genes rather than individual nucleotides. So as I was telling you, exonucleases have been ruled out, they're of no use. We need to find some other types of enzymes for this genome digestion step. Now, who found out these enzymes for us? Who found out these enzymes for us? Arbor. Arbor is the name of the scientist who actually worked on bacteria. What aspect of bacteria? Arbor worked on bacteria's defense mechanism. Arbor worked on bacteria's defense mechanism against the bacteriophage viruses. You know bacteriophage viruses, the viruses that infect the bacteria. So Arbor was a person who was actually working on the bacteria, on their defense mechanism. He was studying the defense mechanism of bacteria against the bacteriophage viruses, and he noticed something interesting. He noticed presence of an enzyme system. That means a group of enzymes basically in the bacteria. He noticed an enzyme system in the bacteria called the restriction methylation system. Called the restriction methylation system. Arbor was working on bacteria's defense mechanism against bacteriophage viruses, trying to understand it. He noticed presence of an enzyme system in the bacteria called the restriction methylation system. What exactly was this system? In this system, Arbor found out that bacteria use an enzyme called methylase enzyme. Bacteria use methylase enzyme to methylate every nucleotide of their own chromosome. The bacteria use methylase enzyme to methylate every single nucleotide of their own chromosome after the entry of bacteriophage. See, we have studied human health and disease, the immune system in detail, how the immune system acts when the pathogen enters your body. There are APCs, the antigen presenting cells, engulfing the pathogen, and once they engulf the pathogen, they are going to present it to the lymphocytes. The lymphocytes which get exposed to the presented antigen will become activated, will become stimulated, and then interleukins are going to give them co-stimulation, that is activation of the lymphocyte, and that is when your lymphocytes start acting against the pathogen. So it's something similar over here also. Once the virus is inside the bacteria, what is the bacteria supposed to do? Try and protect itself. So how does it protect itself against the virus? First thing that the bacteria does is uses methylase enzyme to methylate nucleotides of its own chromosome. Okay, and then the bacteria produces the other set of enzymes. The bacteria produces another type of enzymes called endonucleases. Look at the word endonucleases. What was our pancreas producing? Exonucleases. What are the enzymes that the bacteria are producing? Endonucleases. The bacteria produces another type of enzymes called endonucleases, which do not bind to the host bacteria's own genome. Why? As it is protected by methylation. The endonucleases do not attach to the methylated genome, and whose genome is methylated? The bacteria's own genome. So endonucleases do not bind to the host bacteria's own genome, but will attach to the viral genome. Why will they attach to the viral genome? Because the viral genome is unmethylated. Because the bacteria methylates only its own genome, not the viral one, obviously. So the endonucleases will attach to the viral genome and will attach only at a fixed or a particular location. When we are saying that endonucleases will attach to the viral genome, does that mean that the endonucleases will just attach anywhere randomly wherever they want? No, even the attachment follows a rule. What is that rule? The endonucleases will attach, number one, only to the viral genome. Number two, only at a fixed or a particular location, which is called a palindromic recognition site of the endonuclease. Okay, so the bacteria produces endonucleases. They do not bind to the host's own genome as it is methylated, but will attach to the viral genome because it is unmethylated. And where will they attach? Randomly anywhere? Of course not. They will attach only at a fixed or a particular location called a palindromic recognition site of the endonuclease. Now, one more thing before we go further. A single bacteria can produce many endonucleases. There's no restriction on the number of endonucleases that a single bacteria can produce. A single bacteria may produce 10 endonucleases in response to the entry of the viral cells in it. And each endonuclease enzyme has its own particular fixed recognition site. Okay, the point to understand over here is, let me show it to you in a diagrammatic form. Here is a bacteria, and here's the bacteria's chromosome. Here's a phage virus that has decided to infect this bacteria, and once the phage virus infects the bacteria, what is it going to do? Send its genome inside. This is what Arbor was studying, that what will the bacteria do now? So it's a very simple thing. What the bacteria does is produces methylase enzyme, and methylase enzyme we can notice is adding methyl group to what? The bacteria's own chromosome. The chromosome is methylated by methylase enzyme. But is the viral genome also methylated? No. So now the bacteria produces certain enzymes called endonuclease enzymes. This here is an enzyme produced by the bacteria. What type of enzyme is it? Endonuclease. The endonuclease enzyme does not attach over here because it is methylated. What is the only target of the endonuclease? Viral genome. So what the endonuclease does is it is supposed to basically attach to what? Viral genome. And if it attaches to the viral genome, let's say over here like this, what is it going to do to the viral genome? At the place of attachment, cut it, so that the viral genome is converted into what? Two pieces. Maybe the enzyme then shifts here and attaches over here, cuts it. So now the viral genome is cut into what? Three pieces. So do you think the virus is going to grow inside the bacteria now? Of course not. So what are endonuclease enzymes? They are the defense mechanism of the bacteria against the virus, because they can digest the viral genome. Can they digest the host's own genome also? No, because that is methylated and protected. But can they digest the viral genome? Yes, that is what they are meant for. Okay, a bacteria can produce many endonucleases. Yeah, one more thing. This is one endonuclease that has been produced by the bacteria. It attaches to the viral genome over here and cuts it. At the same time, is it compulsory that the bacteria will be able to produce only one endonuclease? Of course not. The bacteria may produce one more endonuclease with maybe a different type of shape. So as I've told you, the same bacteria can produce endonuclease enzymes of many types. Here is another endonuclease produced by the same bacteria. What about this endonuclease? Even this is supposed to do the same thing. Can it attach to the host genome? No, because that is methylated. Can it attach to the viral genome? Yes, it is supposed to do that. And break down the viral genome into what? Fragments or pieces. Okay, but then the situation is, in this case, a single bacteria has produced how many endonucleases? Two. Is it possible that both the endonucleases can go and compete with each other to try to attach to the same place on the viral genome? Not at all. There is no competition ever between endonucleases with respect to their site of attachment on the viral genome that they need to digest. And why is there no competition between the endonucleases ever? Because each single endonuclease that is produced by the bacteria has got its own single, particular, fixed, or we can say unique recognition site, which is completely different from the recognition sites of other endonucleases. That's a keyword over here. A bacteria can produce many endonucleases. Now the point is, if all the endonucleases want to attach to the viral genome on the same place, there's no point. They'll be just competing with each other only. So rather than doing that, the bacteria produces many types of enzymes. One enzyme attaches over here, cuts it. At the same time, the bacteria produces one more enzyme, and this enzyme may go and attach somewhere over here. And once this attaches over here, it introduces a cut here also. So simultaneously, since the bacteria produced two enzymes at the same time, both of them will go and attach to the viral genome at different locations. Okay, once they attach to two different locations, they might induce cut at the same time, cutting the viral genome into how many pieces immediately? Into three pieces. So a bacteria can produce many endonucleases, and each endonuclease enzyme has got its own particular fixed unique recognition site on which it can attach, and if this site exists on the viral genome. So this is also an important consideration, right? What if the bacteria produces an enzyme but the viral genome doesn't have a recognition site for that enzyme? Do you think this enzyme will be of any use now? No, it will be useless. The bacteria produces an enzyme, listen to this again. The bacteria produces the enzyme, but the enzyme fails to find out any recognition site for itself on the viral genome. So such enzyme is a waste. They'll just keep lying down in the cell. But then it's okay, it doesn't matter. They might be helpful in future against maybe some other type of virus. So a bacteria produces many endonucleases. Each endonuclease has its own particular fixed unique recognition site on which it needs to attach, and if this site exists on the viral genome, then only the enzyme, the endonuclease enzyme, will attach to the viral genome and digest it. Now what about digestion also? If the recognition site for the enzyme exists on the viral genome, the enzyme is going to attach over here and will digest only one phosphodiester bond on each strand. The endonuclease enzyme digests only one phosphodiester bond on each strand. Compare this with what we saw in the exonuclease. The exonuclease was digesting each and every single phosphodiester bond, producing what? Nucleotides. What we are understanding over there now is an endonuclease enzyme comes and maybe attaches over here. And once the endonuclease attaches over here, it cuts this phosphodiester bond. If this phosphodiester bond is cut, I am getting 1 and 2 fragments of the genome. Okay, so restriction endonucleases, number one, they don't just attach randomly anywhere they want. They need to find a particular recognition site, and that recognition site is always a palindrome. I hope you understand palindrome, right? For example, let's say if this sequence here is GAATTC. Look at the complementary strand. What will be the sequence? GAATTC. So this kind of sequences which are same from both the sides. 5 to 3 GAATTC, 5 to 3 GAATTC. What is this? Palindrome. This is the site on which enzyme can come and attach. Okay, so if we are actually talking about all the steps that we have done today, number one, we have the DNA precipitates in a test tube. Where did I get these DNA precipitates from? By spooling from that test tube where the genome isolation experiment was done. Then once I have these DNA precipitates over here, I'll basically dissolve them in small amount of buffer, and I need to basically digest them. To digest them, I need enzymes. One choice that I explored was my own body, pancreas, that produces nucleases. But the result is when you add nucleases over here, this is what you get. So we are not going to use these enzymes. The use of pancreatic enzymes is ruled out. So then we need some other enzymes that can attach somewhere in between, cut somewhere in between, produce what? Fragments instead of nucleotides. So these enzymes now that are going to attach to the genome somewhere in the center, somewhere in between, not exactly on the edges. What are these called? Endonucleases. Now where were the endonucleases found from? This is what we are discussing here. Arber was a scientist who was working on bacteria's defense mechanism against bacteriophage viruses. He found out that whenever a phage virus enters the bacteria first of all methylates its own genome with the help of methylase enzyme, and then the bacteria produces certain enzymes called the endonucleases that will attach at fixed locations, unique locations on the viral genome, cutting it into pieces. If the viral genome gets cut into pieces, will the virus be able to grow inside this bacteria? No. So what have the endonucleases done? The endonucleases have succeeded in restricting the growth of the virus inside the bacteria, which is why a complete term for the endonucleases is restriction endonucleases, RE. Okay, so endonucleases will attach to fixed recognition sites, which are palindromes on the DNA, and digest only one phosphodiester bond on each strand, so that the genome gets digested or broken into gene fragments and not individual nucleotides. And isn't that what the bacteria wants? Because it wants to get rid of the virus. Whose genome are we talking about here? The viral genome gets digested or broken into gene fragments, and but not the nucleotides. So the growth of virus in the bacteria is restricted or prevented. The growth of the virus in the bacteria is restricted or prevented. Restricted or prevented by what? By the action of endonucleases. How? Because they digested the viral genome into pieces. They digested the viral genome into pieces. Hence, endonucleases are also called restriction endonucleases. Why are they called endonucleases? Because वो edges पे attach नहीं कर रहे, बीच में कहीं पे attach कर रहे हैं अंदर. That's why endonucleases. और ultimately achieve क्या कर रहे हैं? They are restricting the growth of the virus inside the bacteria. So what is the name? Restriction endonucleases. Okay. So this was the work done by Arber when he was trying to study the defense mechanism of bacteria against the bacteriophage viruses. And that's it. This was the idea. So Arber's discovery of this defense mechanism in the bacteria. Arber's discovery of this defense mechanism in the bacteria paved the way or showed the way for what? Obtaining such enzymes from the bacteria, obtaining such enzymes from the bacteria and using them in laboratory to digest the genome of our desired organism, which was producing our desired product but for its own self, not for us, into fragments or pieces. If I can take out a restriction endonuclease from a bacteria, and put it in that test tube where I have the genome, will the restriction endonuclease work in the same manner as it works in the bacteria cell? Yes. And will it break down my genome into fragments or many pieces? Yes. By attaching to recognition sites, which are palindromes, the enzyme is going to attach to my genome and break it down into fragments. Here's your test tube where you have the genome dissolved in small amounts of buffers. When I add restriction endonucleases, RE for short, when I add restriction endonucleases in this test tube, what are they going to do? They're going to attach to this genome. They're going to attach to this genome on what? Palindromic sites, and then cut a phosphodiester bond inside those palindromic recognition sites, producing what? Fragments. Fragments of this genome, and that is what we wanted. Okay. So where are these restriction endonucleases obtained from? Bacteria. Clear? So Arbor's work paved the way for obtaining these enzymes from the bacteria. RE from the bacteria. Why do the bacteria produce them? As their natural defense mechanism against viruses. But then if bacteria are producing them, we can always get them from the bacteria. And once we get them from the bacteria, what will we do? Put them in the test tube to digest the genome. Will the enzyme be able to attach to the genome? Yes, if the recognition site is there. The recognition site, by the way, as I've told you, is always a palindrome. So if the enzyme finds a recognition site for itself on the genome, it is going to attach to the genome. And once it attaches, it cuts the phosphodiester bond, and that's it. Produces what? Fragments. So restriction endonuclease from the bacteria are added in the test tube, having the genome that we extracted from the organism who is the natural source of our desired protein product. Okay, restriction endonuclease from the bacteria is added in the test tube, having the genome. Which genome? The one that is extracted from the organism, who is the natural source of our desired product. Once I do this, the result will be, here's your test tube. What did it have initially? In the buffer solution, DNA precipitates. But now what will it have? Smaller fragments of the DNA. These are now the digested DNA fragments. And as I told you, this is what I wanted. I can basically take out these digested DNA fragments now and somehow identify that particular fragment which is going to code for my product. So use that fragment, put it in some other cell and get the products. That's it, done. Okay. So what we learned here was that if I have obtained a genome, from what? From an organism who naturally produces that product which I want at a commercial scale. So I just take out the genome of that organism and I want to cut or digest that genome and obtain that gene. I tried exonuclease enzymes, but they did not work because they broke down the genome into nucleotide and I couldn't do anything with those nucleotides. So then I had to find some other enzymes. Obviously, some research was required for that. So Arbor was researching on certain enzymes found in the bacteria as a natural defense mechanism against viruses. He found out that restriction endonucleases are the enzymes which are produced by the bacteria when the viruses enter in them. And these enzymes will be attaching to the viral genome on a particular fixed recognition site, which is always a palindrome. And once they attach, they are going to basically digest the phosphodiester bond at that place where they attach, breaking down the genome of the virus into fragments. And that is the type of enzymes that I also want. I also want to basically break down my genome into fragments. So I should be using these enzymes, and then I am using these enzymes. So what are the enzymes that are of choice, are that are preferred enzymes in biotechnology? Restriction endonucleases. Why are they called restriction endonucleases? Endonucleases because they are attaching somewhere in between. They are attaching maybe somewhere over here, maybe somewhere over here, maybe somewhere over here, and cutting it, not on the edges. But why restriction word? Because in the bacteria, when the bacteria is producing them, who are they restricting inside the bacteria cell? The growth of the bacteriophage virus. So since they are restricting the growth of bacteriophage virus, restriction enzymes, since they are cutting the genome of the virus by attaching somewhere in between, not at the edges, endonucleases. So finally, what have I got? Digested genome, DNA fragments. So the idea that such enzymes exist in nature was initiated by Arbour. There are two more scientists whose names are worth mentioning here, Smith and Nathans. What they did was Smith obtained HIN2. HIN2 is the name of HIND2 is the name of the first ever restriction endonuclease. Used in laboratory for this purpose, for digesting the genome of the organism. Okay, and Nathans demonstrated or developed the protocol. Demonstrated how to use the restriction endonucleases in laboratory. See, ऐसा नहीं कि आपको बस restriction endonuclease हो तो एक चम्मच भर के या एक स्पून भर के डाल दिया. Not at all. There is always a fixed protocol, and the protocol is you need to basically add just a fixed quantity of the enzyme in a fixed amount of DNA at a fixed osmolarity temperature. For how much time are you basically supposed to give the exposure to the enzyme? So all that protocol was developed by Nathans. Okay, so their contribution also is equally important in developing the knowledge about restriction endonucleases. Okay, so this is the end of the lecture today, guys. We have basically learned today that the genome which I obtained from the organism who's the natural source of my desired product, I have to digest that genome. To digest that genome, the best enzymes are restriction endonucleases. Restriction endonucleases are naturally found in bacteria, and we can obtain them from the bacteria. We can use them in the laboratory. What will we do with them in the laboratory? Just put them in the test tube, let them do their magic. What is the magic? They'll find some palindromic recognition sites on this DNA, attach to those sites, cut those sites, break down the genome into fragments. Why is the virus producing them? To do the same thing against the bacteriophage viruses. Why are we using them? Because we want to do the same thing, break down a genome into gene fragment in the laboratory. Okay? There's more to restriction endonucleases. There are types of restriction endonucleases, type 1, type 2, type 3. Okay, and then within type 2, that is the preferred enzyme, there are more variations. So we'll basically take forward those variations of restriction endonucleases in the next class. That is all for today. Okay, so keep studying, keep spreading the word about the channel to your friends and acquaintances or whoever, and I'll see you in the next lecture. Thank you everyone. Bye bye.